everyone, for one last time this semester. My name is Marissa Botello, and welcome to my Week 8 presentation, recapping what we've learned in the past few weeks of labs about ATP synthesis and nucleotide production, which can ultimately be used for DNA replication. To cover this, we'll be looking at the 15 pages of metabolic pathways we've built into this poster. Metabolism is the set of chemical reactions that occur in a cell which enable it to keep living, growing, and dividing. Metabolic process can be catabolic, which is obtaining energy and reducing power from nutrients, or anabolic, which is producing of a new cell component through processes that require energy and reducing power obtained from nutrient catabolism. There are a large number of metabolic pathways constantly occurring in our bodies, some of the most important being glycolysis, the citric acid cycle, oxidative phosphorylation, the pentose phosphate pathway, the urea cycle, fatty acid beta oxidation, and gluconeogenesis. And although for explanation purposes today it will sound like these things are happening in a line, kind of one after another, it's more like these are constantly cycling. The body is never not metabolizing. The major currency for human metabolism is glucose. It's our universal fuel for all of the cells in our body to derive energy from. In mammals, glucose is the only fuel that the brain uses under non-starvation conditions, and the only fuel that red blood cells can use under all conditions. Although many carbohydrates exist for us to consume, glucose is the most prominent monosaccharide for metabolism of most organisms. We aren't really sure why evolution selected glucose, but the theories behind that mostly have to do with the availability in primitive biochemical systems. Also, because glucose has a strong tendency to exist in ring formation, it has a lower tendency than other monosaccharides to non-enzymatically glycosylate proteins by first forming shift bases with their amino group. As for what we do with glucose once entered our bodies, glucose is a reduced compound from which electrons are harvested by oxidation reactions. Electrons are captured by electron carriers and, when mitochondria and sufficient oxygen are available, transferred to the electron transport chain, where they are ultimately donated to molecular oxygen with the formation of water and a relatively large amount of ATP. The complete oxidation of glucose is achieved by the process of glycolysis, which yields a relatively small amount of ATP and electrons, and the reactions of the citric acid cycle, which also yield a small amount of energy but a larger amount of electrons. Glucose carbons are oxidized to carbon dioxide and largely released into the environment. A second pathway, the oxidative phase of the pentose phosphate pathway, partially oxidizes glucose to obtain electrons that are used for reductive biosynthetic reactions and to prevent or reverse oxidative damage to cells. In this pathway, glucose is oxidatively decarboxylated to yield 5-carbon sugars called pentoses. That may be used in the non-oxidative phase of the pentose phosphate pathway for nucleotide synthesis or returned after a series of interconversions to the glycolytic pathway. Now that we've covered a bit on glucose and metabolism, I want to review the sheets we've put onto this poster and what pathways they show to make my later explanation easier. So I'm going to just start at the top here and just point out what we're looking at in that order before I go into the actual discussion on ATP synthesis and nucleotide building. The first corner shows the pentose phosphate pathway, responsible for synthesis of pentoses and release of the reducing power needed for anabolic reactions. Next is the breakdown of lactose and galactose for use by glycolysis. Then we have fatty acid beta oxidation, responsible for fatty acid breakdown into acetyl-CoA, which is then used in the citric acid cycle. Next up is purine and pyrimidine synthesis. And then finally on our row we have uridylate and inosinate acting as precursors to nucleic acid formation. Starting at the left of the second row here, this page shows chemiosmosis, which is the action of ATP synthase in the proton pump. This is sort of coupled with the page below it right here, which shows the electron transport chain. And together we can refer to these two pages as oxidative phosphorylation, which is the disposal of electrons released by glycolysis and the citric acid cycle, Much of the energy released in this process can be stored as ATP. Moving back to where we were, the second page of this row shows glycolysis, which is our all-important pathway where glucose oxidation occurs in order to ultimately obtain ATP. Glycolysis is the transformation of glucose into pyruvate. And the next page over shows the opposite. We have gluconeogenesis, and this is where pyruvate is converted into glucose. This is glucose synthesis using small precursors, mostly to be used by the brain. Next, we see generation of isoprenes, which will be used in cholesterol synthesis. And the last page on this row, we see generating of serine and glycine from the glycolysis product 3-phosphoglycerate. We also have the generating of arginine, which involves the glycolysis or beta oxidation product acetyl-CoA and the citric acid cycle product glutamate. Tucked in here is also, right here, a really primitive summary of the urea cycle, which is the disposal of ammonium ion in less toxic forms. For our bottom row, we're starting again by looking at the page with the electron transport chain, and since we've already talked about oxidative phosphorylation, I'm going to move on to the next one. This shows the pyruvate dehydrogenase complex on top and the citric acid cycle on the bottom half. The citric acid cycle is responsible for acetyl-CoA oxidation in order to obtain energy, electron carriers, and other valuable intermediates. On this next page, we're showing amino acids made from those citric acid cycle intermediates. Moving on, we see cholesterol synthesis from the isoprenoids we generated above, right there. And on our final page, last on the poster, we have generation of cysteine, which forms the byproduct succinyl-CoA, which moves back and is used by the citric acid cycle. So now I'm going to move more on to those prompt-focused questions, starting with glucose, which I circled in red. Starting with glucose in glycolysis, we're going to talk about how ATP is generated. Glucose is circled there in red at the start of glycolysis, which yields pyruvate. This is the first step in ATP production, as glycolysis generates some ATP and NADH. Both of those are circled in red, and then minus the ATP invested in the beginning. But most importantly, the two molecules of pyruvate move on to the pyruvate dehydrogenase complex. So we're going to follow the arrow. And we see that pyruvate can go through the complex, generating acetyl-CoA, which I've also circled in red, and generating some more NADH and carbon dioxide, which we're going to use later on. The acetyl-CoA generated here, so go ahead and just move down again, enters the citric acid cycle, which generates more small amounts of carbon dioxide, NADH, and FADH2. And follow these red arrows over. I'm going to show you both of the pages here. We can follow the red arrows again and see those electron carriers move to the electron transport chain and help to generate a proton gradient, which drives ATP synthase and the proton pump. Ultimately, oxidative phosphorylation is the largest ATP yielding step. NADH and FADH2 generate 2.5 and 1.5 ATP respectively through this process. So in total, we make 30 ATP for every one glucose molecule that we consume. For the next part of the prompt, I'm going to go on to how nucleotides are made. So we'll start actually back at our glucose in glycolysis, and we see that this can be converted by hexokinase into glucose 6-phosphate. I have that circled in orange right beneath. Glucose 6-phosphate, then we can follow this arrow up. We follow this arrow and we move to the pentose phosphate pathway, where it is converted to ribulose 5-phosphate, and then ribose 5-phosphate, which is a key component in the development of PRPP. Follow this long orange arrow again. We're over here now. We see that PRPP is the start of the pathway which leads to purine synthesis. This pathway converts PRPP through a long series of reactions into inosinate, which can be converted into ATP and GTP, which develop the purines adenine and guanine. So we'll see PRPP moving down to inosinate, generating that ATP and GTP, and ultimately adenine and guanine. And then backtracking on the paper a bit here, because we've made those, we're going to move on to pyrimidine synthesis. We see the carbon dioxide here, which was one of our given kind of start spots. So we'll see carbon dioxide there circled in orange, and that CO2 leads to carbamic acid, which ultimately leads to uridylate. And then we'll move over. If we follow that arrow, we see uridylate can be turned into UTP, CTP, and TTP, which develop the pyrimidines, which will give us uracil, cytosine, and thiamine, respectively. So through these pathways, we can see that glucose leads to ATP synthesis, as well as nucleotide synthesis. I'll back up to show you my poster again here. Thank you, everyone, for watching. Hope you all enjoyed this course, and best of luck.